Thank you very much. I'm Gemma Molinos from the Institute of Psychiatry at King's College London, and I'm delighted to introduce Sam's talk this morning. Sam started his studentship in October 2020, in the midst of it all, so the main project was stopped at that time, and he's used this time really to train in the methods involved. It's quite a brain imaging heavy project that he'll explain in a minute. And he's also just finished writing a comprehensive literature review relevant to his topic. So over to you, Sam, and I hope you all enjoy the presentation. Thank you.
All right, thanks very much, Gemma. Can everyone see my screen okay? Can you hear me okay?
Yes, we can.
Okay, great. So I'm just going to launch right in talking about my PhD project, which is the neuromechanisms of positive symptoms in first episode and prodromal psychosis. So just a quick outline. First, I'm going to give a bit of a background of what that actually means, some of the key questions that come from that, how we're trying to investigate those questions, what we think is going to happen, and then touch on the progress that I've made so far in this first year and a bit. So what is psychosis? Well, psychosis is a very severe mental state, and we tend to categorize psychosis by clustering it around these different symptom categories. And I'm going to be focusing mainly on the positive symptoms because it's most relevant to my PhD. That includes delusions, hallucinations, persecution, things of this nature. But just to acknowledge as well that psychosis also includes these other symptoms, these cognitive deficits and these negative symptoms. Now, psychotic disorders usually have an onset in early adulthood, affect roughly 1% of the population, and there's really high costs associated with them, both for the outcome of the individual, but then also for the societal costs. So as I start to talk about, the neural mechanisms of psychosis, don't worry too much about remembering this brain region doing this and that sort of thing. But what I'd hopefully like to communicate is just the logic of how we're trying to investigate these questions. So the positive symptoms of psychosis at the moment, currently the best explanation for why these symptoms arise is because of dopamine dysregulation. Dopamine is probably a neurotransmitter that you've heard of. It's involved in reinforcing signaling. And what we think is going on in psychotic symptoms, positive psychotic symptoms, is there's an aberrant salience. There's irregular dopamine reinforcement, and that's causing people to pay more attention to things that they shouldn't be paying attention to in a very basic sense. And currently, the way we treat psychosis is with antipsychotic drugs targeting this dysfunction. And that's pretty good for a lot of people at reducing the positive symptoms, but it also doesn't work for a lot of people. People might still have persistent symptoms, lots of nasty side effects. So dopamine isn't the entire story here. And so what we're trying to investigate is really things that are happening upstream in the brain, rather than focusing on the dopamine dysfunction, focusing on other regions that might be contributing upstream. And two of these regions are the prefrontal cortex and the hippocampus, which I'm going to pay the most attention to. And so the way that these brain regions communicate to each other is a balance between excitatory and inhibitory signaling. We have positive excitatory pyramidal cells and inhibitory GABAergic, predominantly, interneurons. And the balance between this excitatory inhibitory firing is really important for brain regions to talk to each other effectively. And one of the things that we think is happening in psychosis is that there is an imbalance between the signaling. One of the reasons why this might be is what we call oxidative stress. So anytime you're using energy in your body, there are byproducts to that reaction. You've probably heard of free radicals, you've probably heard of antioxidants, and free radicals or reactive oxygen species, our body does a pretty good job of mopping these up after we use energy. But if certain regions become hyperactive, become more metabolically active, there's more byproducts, and these aren't necessarily taken care of. And the cells that are most vulnerable to this sort of dysfunction are these inhibitory cells, which are really important at coordinating these neural firings between the different regions. So what we think is happening in psychosis is oxidative stress is damaging these interneurons, causing an increase in activity in the hippocampus, and then increase in activity in downstream regions and aberrant salience. So the key questions really for my PhD is, can we find signs of the hippocampus being hyperactive in psychosis? Can we find signs that there's an imbalance between excitatory and inhibitory signaling? And can we find signs of redox imbalance or oxidative stress? So now I'm going to shift and talk a little bit about the progress made so far and the methods that we're using. So my project is part of the larger stress gap project being conducted by my lab. And we're looking at really at three patient populations, well, three populations: healthy control participants, people at clinical high risk for psychosis. These are people that don't have a psychotic disorder, but they do have attenuated positive symptoms. So we think that they're at the highest risk for somewhere down the line developing a psychotic disorder. And then we're also looking at people experiencing first episode psychosis. These are people that are currently have positive psychotic symptoms. And the procedure goes like this. I've embedded myself within different teams within South East London and early intervention services, and I speak to the care coordinators there about people that may be interested in participating in research. Having done this now for a year, I can say that people want to participate in this research for all sorts of different reasons. Some people just like a little bit of extra money. Some people are really interested in just trying to know what's going on with them and trying to understand a little bit better the changes that they're going through and the stressful experiences that they're going through. And other people, which is really interesting, it has been interesting for me, is just people want to help, even if it's not benefiting them directly. They want to provide researchers with more knowledge so that these problems that they're going through might be better dealt with people in the future. And I think that's really powerful. So once we've identified people to come in and go through the rest of the procedure, we get them to come in, we do some clinical questionnaires, we do some cognitive assessments, and we take a blood test as well, which allows us to take a little bit of genetic information, which we're going to look at later relating to oxidative stress, but I'm not really going to talk too much about that today. And then we get them to do a PET MRI scan. And the MRI aspect of the scan is really the important bit for me, but just to acknowledge as well that we do also do a PET scan at the same time. So it's a little bit more of an invasive scan than some people are used to. So it's really amazing that they're willing to go through this process. And we're normally done within 2 to 4 weeks. So as I said, I'm really using these two MRI-derived component methodologies. One is arterial spin labeling, which just gives us a resting metabolism of the hippocampus. So that's going to show us whether we find signs of hippocampal hyperactivity. The other is what's called magnetic resonance spectroscopy. It's a slightly different application of the magnetic resonance phenomena. It allows us to look at the concentration of a particular metabolite that we're interested in our case, this GABA, this inhibitory neurotransmitter, and glutathione, which is the body's main antioxidant. And we're able to look at the concentration of those metabolites in a particular region. And you can see that's an image from one of the participants that we have there. So what we think is going to happen is in the early psychosis group and also in the people at highest risk for psychosis, we're going to find signs of hippocampal hyperactivity. We're going to find signs of this excitatory inhibitory balance being abnormal. And we're going to find signs that the redox balance of these patients is abnormal, they'll have less glutathione. So other than the participant recruitment that we've gone through, which you sort of flash past it a little bit, but you can see how many we have in each group so far. Obviously, recruitment has been slowed down because of the pandemic, but we are in full swing at the moment, and things are going. We're hoping that we'll be all done in time for me. Other than that, I've been going through various training relating to the methodologies, and also writing this literature review that Gemma mentioned. So this is something I've just submitted, and it's looking at the neurodevelopmental trajectory of the hippocampus and why that region in particular might be vulnerable to dysfunction and the sort of dysfunction that we see in psychosis. I then talk about the hippocampal abnormalities that we find in psychosis and those at risk for psychosis, before talking about the ways in which those abnormalities might be targeted by specific interventions. So you can see from this graph, antipsychotic interventions come quite late after someone's already experienced first episode psychosis. And so if we can focus on earlier treatments, we might be able to attenuate or prevent entirely the development of psychosis. And that's really why this research is important, because if we're able to uncover more about these neuromechanisms, we might be able to come up with better treatments and be able to intervene earlier. So yeah, I'd just like to acknowledge Mental Health Research UK and my lab and my supervisor. And yeah, that's it.
Great, thank you very much. Could you take your slides down?
How do I stop? There we go.
Good, so we've got some questions. Trevor.
Oh, yes, nice presentation. I wondered whether you're able to also quantify glutamate, because you were talking about the excitatory inhibitory balance.
Yeah.
But you're only looking at one part of that, which is the GABA levels.
Yeah.
Are you measuring at 3 Tesla or what?
Yeah, exactly. At 3 Tesla. We do have another MRS voxel in the same region that the one I sort of showed in my slides is. The sequence that I'm mostly focusing on for my PhD is looking at GABA and glutathione, but we do have a press voxel in the same area or a very similar area so we can get a measure of glutamate as well there.
Okay, thank you.
Thanks, Trevor. And James?
Hi, Samuel. Thanks for the talk. It was really interesting, and I wish someone had explained the sort of basic neurobiology of how dopamine and the hippocampus and the striatum and so forth fitted together when I was a student, because it's something I've always struggled with as an epidemiologist. So my question was really just a bit of clarification about that. So it makes perfect sense that you said that you want to look at kind of upstream things, upstream of dopamine, but presumably if you find that there are these changes in the hippocampus that are important in psychosis, then they don't necessarily even need to go through the dopamine system. Is that right?
Yeah, that's absolutely right. So, I mean, dopamine is part of the picture for some people, it seems, but it's not to say that it's the whole story at all. Because hippocampal abnormalities, part of the reason why we're looking at 3 groups and people not just having psychosis, but people before the development of psychosis is, if we can find abnormalities in these systems that, are prior to any dopamine abnormalities, we can better characterize how that might be targeted earlier. And, there are many other interpretations that of all positive symptoms, but particularly the negative symptoms in other aspects that don't have anything to do with dopamine, but may still have something to do with hippocampal hyperactivity.
Right. Great. Thank you very much. I really look forward to seeing the results as they come out in the next couple of years.
Yeah, good luck with recruitment. We've got 2 more questions. Juliana's first, then Laura.
Thank you. Great presentation, Samuel, so it's really nice. I'm always really interested in these types of studies. I say these types of studies, you always get different groups, and just because you'd mentioned before, which I thought was really helpful, about the different reasons why people get involved in research. And nothing else to do, or money, all those things. But I was just wondering, in terms of I don't know, the user voice, in terms of just the area that you're focusing on and what they think about that.
Yeah, I mean, it's really tricky, the way that we recruit is primarily through speaking with care coordinators, and it can be really tricky, I guess, to get direct input from patients. As we communicate about the study, every individual client will have different concerns, different things that they raise, different ways of thinking about what's going on for them. Psychosis is one label that we use, but often I find, speaking to the patients, it's more useful just talking about the stressful experiences that they're going through now, rather than using these labels. So that's one thing that I've learned, speaking to patients. But yeah, I don't know. That's about all the answer I have. Sorry.
No, it's just that the reason I mentioned it, because I just think people start to make these distinctions and they're somewhat artificial. This is such important research, and the idea about what it can kind of lead to. But I often wonder, or I see it a lot of the times, that even though people might get involved, but they don't always understand why.
Yeah.
What it's about and what you're trying to do. And
we do try to, you know, at many points along the line, really try and outline, you know, as clearly as we can, and we do have criteria around what we believe to be a certain level of capacity for people to be able to consent as well, because it is a little bit of an invasive study. You know, the PET side of things, they have to have an arterial line at the wrist. They're injected with a radio tracer. That can be a quite stressful experience for some people. So, you know, we do try and, you know, outline, you know, many points along the way, what's involved with the study, the benefits, the risks of taking part, you know, and you know, that it's not, this is not a clinical scan. You know, we're not going to be able to tell anything about your brain specifically. You know, we do outline that up front to people.
It is a really important point. Thanks for making it, Juliana. We've just about up to time, but Laura Davidson, can you fit the question, and Sam, your answer in under a minute?
I will do my best. Hi, Sam, thanks again for your presentation. I'm just turning on my video briefly because my connection here is a bit unstable in Africa. My question really, you may not be able to answer it. You said that one of the groups was for people with attenuated positive symptoms, and then there are obviously people with positive symptoms. But the dividing line between the two is a little tricky. How are you deciding on the diagnoses? Also, surely there may be an issue where possibly they go over the line and then they are actually diagnosed as psychotic or having schizophrenia during the trial, and how would you deal with that?
Yeah, it's a good question. I mean, our primary dividing line between these patients is the groups that we recruit them from. So early intervention services in the case of First Episode, and the Oasis service in South London for the clinical high risk. Now, within that clinical high risk service, there is, you know, we administer the CARMS questionnaire to them, and that does have, you know, a level of symptom threshold that would be over our limit for that group, and that I'm not aware of any specific instances in our recruitment that has occurred, but yeah, that is essentially our dividing line. You know, we during the clinical interview stage, we establish, you know, we question them about their experiences and score them based upon that.
Thanks, Sam. I better draw you to a close there. Thank you very much for a great talk.